Hello, my name is Archie, and today I'll be discussing my research project, modeling platelet dynamics in hepatic sinusoid using machine learning and statistical analysis. So first we have to quickly discuss the structure of the liver. The liver is made up of liver lobules, which themselves are made up of hepatocytes. Kupffer cells are the major filters, and they will be ingesting any compounds, foreign compounds, and pathogens. Thrombocytes are commonly known as platelets, which are involved in immunothrombosis and blood clotting. I have 5 different objectives. First, it's data acquisition through intravital microscopy. Intravital microscopy is amazing as it allows for in vivo imaging of live specimens. And then we move on to data cleaning. Since it is in live animal imaging, we have a heartbeat, which distorts our data quality. So we must correct for that. After data cleaning, we move on to machine learning model training, which involves 2 parameters, platelets and the background. This is a supervised model as the user has to do the training. We move on to data processing, in which we import the trained model from before, and then we move on to data analysis and interpretation with Python. So data stabilization, we can see that the red symbolizes the area of Kupffer cells. And so Kupffer cells are known as non-motile cells. So if we stack them on top of one another over time, we expect to not see a huge area. But as we can see in the pre-stabilization, the area is moved and really jagged, whereas in the post-stabilization, the area has been confined a little more. So we know we've achieved some stabilization in our data set, and it can be visualized in these 2 images, pre-stabilization being on the left and post-stabilization being on the right. Now for the machine learning model training, we did 3 different training sessions going from B to D, panels B to D. And we can even label this small tiny area as shown in this example. And you and I can see there's 3 different platelets, but how do we tell the model this? So we can start labeling, and as we increase labeling, platelets being in yellow, background being in blue, the uncertainty, which is in cyan, decreases, and that's predicted by the model. Once we are satisfied with our model, we can apply it and see and visualize different tracks as the platelets travel through different capillaries in the liver called sinusoids. And I compare it to the threshold model here. The threshold model struggles. It has a lower linearity of forward progression, which means it's not able to track cell to cell from frame to frame. We can see the data is jagged, and we can't map out the sinusoids as well. So the machine learning model provides a different type of quality, and we can have more certainty that it's tracking the same cell from frame to frame without making any mistakes. So to conclude, we learned that we have to capture at a lone line average. That means in intravital microscopy, we need to sacrifice quality for the frame rate. So at a lone line average, the laser scans less, but faster. And this is ideal for machine learning models as we need to be better at tracking. Machine learning models can be trained on really small cells. In our case, thrombocytes and platelets are around 2 micrometers in diameter. Machine learning models can be trained on a variety of data. It could be any data set that you import. Future directions hope to investigate pathogen capture of Kupffer cells by injecting probes, tracking the probes and their velocities, and seeing what determines their capture. And finally, thank you for listening. I'd like to acknowledge my entire lab, everyone who's helped me on this project, as well as the University of Calgary, the Schneider Institute for Chronic Diseases, and NSERC.